Hello, my name is Dr. Michael Johnson, and today I'll be presenting Understanding Tissue Clearing and Tissue Clearing Technologies. I am the CEO of Visikol, which is a contract research organization focused on 3D tissue imaging, 3D tissue imaging analysis, and also 3D cell culture models. And today I'll be talking about tissue clearing and how to adopt it into your 3D tissue imaging workflow. Traditionally, for imaging tissues, the way that we've done this for the last 100 years is to take a three-dimensional piece of tissue, such as a biopsy or a mouse brain, and to section it into ultra-thin two-dimensional slices for histopathology. The reason we do this is that tissues are opaque, and we can only image a very small distance into a tissue before that tissue clouds out our imaging and allows us to capture meaningful information from that tissue. The process of tissue clearing refers to reducing the opacity of tissues such that you can use advanced imaging techniques like light sheet to photon or confocal microscopy to see through the entire depth of that tissue instead of being able to use only a single slice. When combined with fluorescent labeling, tissue clearing allows for whole tissue 3D imaging. So instead of getting a few ultra-thin two-dimensional slices from a tissue and having to embed and section and label, we can actually label in 3D and image in 3D through the use of tissue clearing, which allows us an unprecedented view of tissues and to acquire all of the data from a tissue instead of a very small subset of a three-dimensional tissue. When we talk about tissue clearing, it's not always incredibly clear what we mean by tissue clearing. The best way to describe this, though, is to look at your hand. Your hand is not transparent, and the reason it's not transparent, though it's mostly water, is that the cells within your hand have different refractive indices between the proteins, lipids, and cytosol. Proteins and lipids have generally a high refractive index, and the cytosol has a low refractive index, that of water, of around 1.33. This causes light, when it's passing through the lipids and proteins and cytosol, to diffract, to bend. And as we have lots and lots of cells in our hand, the light is being greatly diffracted, and that causes our hand to appear opaque. But if we're to change that refractive index throughout our hand, so to match the refractive index between the lipids, proteins, and cytosol, light would pass through unobstructed. So when we talk about tissue clearing, what we're really doing is we're modulating the refractive index of a tissue. We're doing that through either protein hyperhydration, lipid removal, or just changing the refractive index of the tissue by removing the cytosol and replacing with a high refractive index solution. By doing that, it causes the tissue to become transparent and light passes through unobstructed, and the tissue is what we refer to as clear. There are three primary approaches for rendering tissues transparent. The first of which are protein hyperhydration techniques such as TDE, scale, CDB, cubic, fruit, clear T, and focus clear. There are also solvent-based techniques, which are high refractive index matching techniques. These are tissue clearing reagents based on Visikol's histotechnology, BAB, I3, U, and now V-Disco as well. And lastly, the hydrogel embedding techniques such as Clarity, PACT, and PARS, which are quite popular in the academic literature. But it's very important to point out that every tissue clearing technique has its own unique advantages and disadvantages. There's lots of techniques that are out there in the literature. Dozens and dozens of different types of techniques have come out over the last few years, but it's really important to note that each of them has a specific use case where it is incredibly beneficial and other cases where it's not so helpful. The first of which consideration is compatibility with labels. Not all techniques are compatible with fluorescent protein, immunolabeling, and dyes. It varies greatly based upon the type of tissue clearing technique that you're working with. Also, equipment. Some tissue clearing techniques, like solvent-based techniques, are not compatible with many objectives that are out there. Some are not compatible with plastics that you might contain your samples in, and some are not compatible with polystyrene well plates that you might be doing your processing in as well. Time is very important. Some tissue clearing techniques are very rapid, some are very slow, and this varies considerably amongst the techniques that are out there. But what's very important is preservation of tissue morphology. You can make a tissue transparent, but if you destroy the tissue in the process of making it transparent, you've not preserved the inherent morphology of that tissue, and the results that you've gathered might not be that important for your research question. And then toxicity. Some techniques are very, very toxic to work with, and while they're very efficacious in rendering a tissue transparent, they're dangerous to work with and not suggested. And then lastly, as I mentioned, compatibility with well plates. Well plates are commonly used for automation and 3D cell culture applications, but many of the solvent-based techniques will actually melt right through your well plates. So there's many considerations in adopting different types of tissue clearing techniques. Now, first off are the solvent-based techniques. So we have BAB, we have the discos, and we have Visikol histo. All of these have a high refractive index, which is allowing tissues to be rendered transparent. Some are compatible with immunostaining, some are not. However, fluorescent protein with solvent-based techniques is typically an issue with quenching, so you'll quench that fluorescent protein over time and you won't get enough signal back. Of these solvent-based techniques, they can also alter the morphology of your tissue, and they tend to have a shrinking effect on many tissues from very severe with something like U-disco to not so severe with something like BAB or Visikol histo that primarily preserves the morphology and structure of the tissues. Lipids can also be removed through many of these solvents depending upon how harsh they are, which might not be ideal for your research question. And many of these can be quite toxic and dangerous to work with. So it depends on which one of these you're using, but some of them can be very toxic and some of them can be quite mild and similar to methanol as far as solvent toxicity goes. For aqueous hyper-hydration techniques, these techniques are quite common. They are generally compatible with immunostaining. It varies based upon the technique. They're all pretty much compatible with fluorescent protein, but the problem with a lot of these techniques is clearing time. They can be very slow to clear a tissue, and they can also cause extreme expansion and also cause tissues to become quite fragile, where if you touch them with a forceps, you can actually damage the tissue entirely after weeks and weeks of processing, which is non-ideal. They'll generally preserve the lipids within the tissue, depending upon the technique that you're working with, but they can cause extreme expansion during that process. And generally, they're quite safe to work with the exception of Clear T and Clear T2, which are quite dangerous to work with and are comprised of a teratogen, so definitely not suggested to work with those two techniques. And then probably the most popular techniques are the hydrogel embedding techniques. And when I say popular, I don't mean by use case or number of researchers using these, but one of the first techniques that came out was the Clarity technique in 2012 by researchers at Stanford. And that paper had garnered a lot of support, a lot of views, and a lot of interest. However, these techniques can be very challenging to implement. They're compatible with all labeling techniques, but they can take weeks of time. They require special equipment to actually bring into your lab. They can cause changes in tissue size and shape, and they're also damaging to tissues. So these processes actually remove the lipids from tissues to render them transparent, which, while providing extremely great quality throughout your tissues and great clarity, it causes lots of problems in trying to validate your results because you've totally changed the morphology of your tissue in the process of trying to image it. And one of the challenges with these techniques is that they are quite toxic to work with. We have a lot of acrylamide monomers used in these approaches, which is non-ideal for regular use in your lab. And again, they're very slow, they're hard to work with, but they provide a great level of clarity through a tissue for a low throughput in your research laboratory. Now, to dive into some of the specifics on the next few slides here, we're going to give some case studies looking at the new N labeling with the Abcam tissue clearing kit and showing how to implement this into your research workflow. One of the first things that I wanted to cover here is optimizing tissue thickness and antibody concentration. This is one of the most important steps in adopting tissue clearing into any research workflow, regardless of what tissue clearing technique that you're working with. The reason for this is that many researchers will see one of these papers with a whole brain labeled and imaged in 3D, and they'll want to do that without realizing that that is terabytes of data. It's extreme data processing. It's also incredibly challenging to label an entire tissue and could have taken months, if not years, to get that protocol in place for that single image they see in a paper. And because of that, we have a protocol here for optimizing labeling, starting small, and trying to ensure that you can achieve uniform labeling for your tissue before you move up to that whole brain-sized tissue that you're interested in. And for many questions, going to a very large size tissue is actually non-ideal, which I'll talk about in a moment here. So tissue size, this is the most important consideration for tissue clearing and where we see the most issues with people adopting tissue clearing into their workflow. What most researchers do is they start way too large with a tissue. They spend weeks trying to optimize with their antibody labels, and they spend hundreds, if not thousands of dollars on labels trying to get them to label their large tissue. The problem with this is that tissue labeling time triples when tissue thickness doubles. Therefore, if you go from a 1 millimeter mouse brain slice to a whole hemisphere, the amount of time required to label that tissue piece dramatically increases. What we suggest to pretty much all researchers is to take tissues, such as a mouse brain hemisphere or a whole mouse brain, and to slice it into 1 millimeter thick pieces for imaging and for tissue processing. This dramatically reduces the amount of time required to process your tissue and also allows you to image with traditional air objectives on any confocal microscope. If you go much thicker than that, labeling becomes a tremendous problem and issue and takes a very long period of time, and you also need special optics and special microscopes to even image the tissue if you can label it. So therefore, we practically suggest that researchers pare that desire down to 1 millimeter sections instead of going after a whole hemisphere. This allows for much easier imaging, much quicker studies, and you can digitally reconstruct these sections back together later if your research question requires that, but most research questions do not. Now, the choice of antibody is very important. We have optimized several antibodies with Abcam that allow us to get uniform penetration through 1 millimeter mouse brain sections, as well as 3D cell culture models in their entirety. Not all antibodies are created equal. You should choose antibodies validated for IHC. Different antigens will behave differently. You'll see that some antibodies will label very easily throughout the entire depth of a 1 millimeter mouse brain section, and for others, they won't get that far at all, and you'll really have to optimize the process of getting those labels across your tissue. Now, monoclonal versus polyclonal. Monoclonal has less background, but potentially less signal. Polyclonal, possibly higher background, but of course a brighter signal. And some commercial antibodies do not work well in 3D at all. So GFAP, for example, in some cases, you'll have no signal from one marker you're working with, but excellent staining with another marker, such as Abcam's chicken IgY anti-GFAP antibody. So there's a lot of screening that needs to take place before you can jump into trying to label your entire tissue. And we've worked extensively with Abcam to validate Abcam antibodies for labeling throughout the entire depth of 3D cell culture models and 1 millimeter mouse brain sections. It's an effort up front, but allows you to really adopt tissue clearing and tissue labeling much more rapidly into your research. So a lot of effort should go into choosing an antibody before you dive into trying to label a very thick piece of tissue. Now, antibody concentration is really important. So if we think about going too high or too low, we're going to get not a lot of signal, or we're going to get a lot of signal, but the signal might only be on the exterior of our tissue because that antibody might not be able to penetrate into our tissue because it's blocking itself from getting in there. And I'll talk about this in the next few slides, but you can cause a dense ring around your tissue of staining if you go too high with your concentration, whereas if you go too low, you're just not going to get enough signal back to actually be able to detect it. So it's a fine line behind trying to get lots of signal and also trying to get your antibody throughout your tissue, which I'll talk about in a moment here. Now, antibody concentration is a balancing act. If we go too low or we go too high, we'll have issues with actually getting signal or getting our antibodies stuck on the outside of our tissue, which you'll see in a couple images coming up here. So if I go with my optimal concentration, I'm going to get good penetration through my tissue, but if I go with a concentration that's too high, the antibody will actually block the channels through the tissue, and outside antibody that's not inside my tissue yet will not be able to penetrate into it. I'll get a very dense ring of staining on the outside of my tissue. This is a telltale sign that our concentration is too high for the antibody that we're working with. And with uniform labeling across the entire depth, we'll see that all of our epitopes are labeled quite evenly through the depth of our tissue, whereas again, with a concentration too high, we'll get uneven staining, we'll get some pockets of staining, but we'll get a very dense band of staining outside of our tissue of interest, which is of course non-ideal. We want to have that label go across the entire depth of our model that we're working with. The way we suggest optimizing antibody concentration is to start small. This is really what we tell every researcher is before you dive into that large piece of tissue that you're trying to work with, start small. So cut a few thin sections, so 100 to 300 micron thick sections of your tissue of interest, and process these tissues through our antigen retrieval steps, block tissues in blocking buffer, incubate the tissues with varying concentrations of antibody, and then see what happens. Use an epifluorescent microscope to specifically see how far the staining is getting across these tissues through cutting a vertical wedge across the depth of the tissue and looking to see how far that labeling has actually gotten. And what you'll see is that for certain concentrations, you'll get a dense band of labeling on the outside, but you won't get penetration. For lower concentrations, you might get a more uniform labeling, but that signal is not high enough. So by titrating in small sections, we can see, does our label actually get into the tissues, and can we get labeling across a large depth of tissue with changing the concentration and also potentially changing the incubation time? This will allow you to know very early on that, hey, my antibody does not work at all for thick sections, and I should try a different antibody. And then, as you get uniform labeling at 100 or 300 micron section of your tissue, move up in tissue thickness till you get to your ideal tissue thickness. You'll actually reach a limit where you'll realize that you can't get uniform penetration past a certain depth. And that's really the depth that's limited by that antibody, but you need to find that empirically through studying exactly how that penetration is affected by these parameters. Incubation time changes a lot with thickness, like I mentioned before. So if we're looking at a very thin section, like 100 or 125 microns, it can be about 30 minutes or so to get labeling across the depth of that. However, we're trying to label, let's say, a whole mouse brain or a hemisphere. If that label can get across that entire depth, it could take 40 to 120 hours or longer, and that's with primary labeling. Secondary labeling, of course, is going to double that time to get more signal back to the detector on your imaging system. So really is an advantage of cutting your tissue into smaller pieces, whether it's a mil or 2 mils, and then imaging those and processing those instead of trying to image a whole mouse brain hemisphere or a whole brain. There's very little practical reason to ever try and image a whole brain or a whole hemisphere when smaller sections can be imaged, analyzed, and pieced back together to dramatically save you time, money, and to just reduce the barrier to adoption of tissue clearing and to imaging. So always try and start small with tissue labeling and, of course, tissue clearing. In this next section, we're going to talk about imaging and how to actually acquire data from your tissues that have been imaged in 3D and what that process looks like for choosing the right labels, looking at secondary labeling of our antibodies, and how best to process our tissues with the Abcam and histo reagents and antibodies. So tissue clearing tips, direct or indirect labeling, autofluorescence, multicolor labeling, and troubleshooting. So first off here, direct versus indirect labeling. So with direct labeling, the antibody is conjugated to a fluorescent marker. There's no amplification of signal, so sometimes not sufficient for larger tissues. However, this is faster. So if we're only using primary labeling, we don't have to go through a secondary labeling process, which doubles our labeling time. So to use the extreme example of trying to label a whole mouse brain, that could take 240 hours to actually label that whole mouse brain with primary and secondary. So an incredibly long period of time to do that, whereas if you just had primary labeling, it would be 120 hours. So we're cutting that time in half. However, the problem with primary labeling is that for thicker tissues, you're not going to get enough signal back the deeper you go into your tissue. So you might get labeling to 1 millimeter in depth, but you might only have enough signal back to your detector that's over the noise of the background to get 100 or 200 microns of depth into your tissue. So practically speaking, we always try and go with secondary labeling. Though it reduces our throughput, it dramatically improves our signal intensity and also the quality of the images that we're acquiring from our tissue because we have a much higher signal compared to the background that we're getting back from our tissue. So indirect labeling, it requires 2 different steps, but that signal that we're getting back is much, much better and provides us a much higher quality of image back to our detector. Autofluorescence is always a problem with any kind of fluorescent imaging that you're using because if you have a high background signal on your tissue, it doesn't matter how uniform your labeling is or how strong that intensity is, you aren't going to be able to see it because you cannot break it out from the background. And because of that, your image quality will be very, very poor. We see this commonly for liver tissues, for kidneys, for tissues that are highly collagenous in nature, we get lots of background fluorescence. So to mitigate this, you can bleach your tissues, you can try all of your tissue processing steps at 4 degrees C instead of room temp or 37 degrees C, and you can also image your tissues with a background channel and then subtract that channel out from your other channels through data processing after you've analyzed your tissue. For multicolor labeling, we can typically go after up to 4 targets at the same time, depending upon your imaging system and the filters that you have. You have to choose your labels wisely so you don't have bleed over between the different channels, but generally speaking, you can get up to about 4 channels, maybe 5 channels, depending upon the type of microscope that you're working with and the type of filters that you have on your microscope system. You must choose your primary and secondary antibodies carefully, though. You need different host species for each target. So, example of 2 targets, GFAP and NuN, we'd use a chicken anti-GFAP, Alexaflor 48 conjugated goat anti-chicken secondary, and then rabbit anti-NuN with a Texas red conjugated donkey anti-rabbit secondary. So primaries can be incubated simultaneously, though, so we don't have to do this sequentially and dramatically increase that time. So we can do those at the same time. And then secondaries can also be incubated simultaneously. So we're not having to go from 2 steps to 8 steps. We can do those at the same time, even if we're using 4 targets, so long as we choose what we're doing for labeling very, very wisely. To give you some images here, these are some Z projections of a rat brain to show that you can, in fact, image in multiple channels. And in this case, we're tiling 9 different tiles here to create the image on the left. That's a 20x. On the right there is a 40x. So we can show that we can see through this depth, but of course the depth by which we can image is really going to be dictated by the type of imaging system and the objectives that we have on that confocal or on that light sheet microscope. One of the troubleshooting problems that we see come up a lot, like I mentioned earlier, was uneven staining. So if our staining intensity is way too high on the outside of our tissue, like I'm showing on this 3D cell culture model on the top right here, what this typically means is our antibody concentration is way too high. They're getting stuck on the outside, and we're not getting the ability to penetrate all the way into the interior of that 3D cell culture model. Whereas on the bottom, we have more optimal labeling, and we see labeling across the entire depth of that 3D cell culture model. So you'll see a dense band of labeling on the outside, but no labeling on the inside, or very, very dim labeling. And this is an indication of uneven staining and, of course, not the right antibody concentration. So this is kind of a telltale sign. And with labeling and with tissue clearing, this is the most common problem that we see researchers come up to. And then also, optical attenuation due to absorption of photons by the upper layers of tissues can cause shadowing and tissues or layers of tissue below the top of the tissue to appear dim and not to have a lot of signal on them. What's happening in this case is not uneven labeling, but what's happening is as we're getting through the depth of the tissue, the top layers are actually clouding the bottom layers, and we're not getting a lot of photons back to our detector. So one of the things that you can do with some microscopes to interact with this problem is you can actually increase the laser power as you go deeper and deeper into a tissue. So you're increasing that signal that you're getting back, not because the fluorescent labeling is not there, but because you're not getting the same number of photons back to your detector as you're getting higher in a tissue. So that's another problem that we see. That's not labeling induced, that's more so imaging and optical attenuation induced. Now, to dive into the last section here, we're going to talk about imaging and different types of imaging modalities that are out there and how best to adopt them for using with the Abcam kits here with specific labels and specific targets in specifically brain tissue. So one of the things that we see a lot is that researchers will try and label a tissue, they'll try and clear a tissue, but they won't necessarily know how to actually image the tissue and acquire the data that they're going after. Each imaging system has very specific use cases where it's great to use that imaging modality and other use cases where it's non-ideal, and understanding those is very important in adopting tissue clearing, because you can go through your whole process, and if you go to your imaging core and they don't have the imaging modality that's compatible with the type of tissue prep that you did, the tissue prep that you did was not worthwhile. So to dive into this in some detail here, when we look at light sheet or confocal or multi-photon microscopy, they all have different use cases. For light sheet microscopy, generally speaking, what it's best for is imaging tissues that are large and imaging those tissues relatively quickly at lower resolution. So we're trying to image a whole mouse brain intact. The approach that we would take is likely light sheet microscopy to do that. You could try and do it with confocal microscopy and dipping objectives, but really the easiest way to do it is to go ahead and do it with light sheet microscopy. However, the problem with light sheet microscopy is that sample prep can be more challenging than it is with confocal microscopy to really match the refractive index, to find a light sheet microscope as they're not as common as confocal microscopes, and to adopt that into your research project. With confocal microscopes, they're much more ubiquitous. Pretty much every imaging core out there is going to have a confocal microscope that you can use. And confocal microscopes are really best for higher resolution 3D tissue imaging, but smaller volumes. Confocal microscopy also has a tendency to quench fluorophores much more easily than light sheet microscopy because we're illuminating through the entire depth of a tissue, and therefore generally it's better for thinner tissues than light sheet microscopy, but we're going to get better, crisper images from confocal microscopy. So it's a question of throughput, and also a question of what access do you have at your university or at your company for imaging modalities. If you have an inverted microscope, for example, a confocal inverted or an upright, that'll also change some of the capabilities of what you're able to do. An inverted microscope will only be able to image to a depth of about 1 millimeter into a tissue slice, whereas an upright microscope with dipping objectives can get many millimeters into a tissue depending upon which objectives that you have. So there's a lot of considerations here, but first off, before you dive into any tissue clearing project, you have to look at what types of resources you have access to and see what they allow you to do, because some microscopes will limit what you're able to do as far as tissue thickness, and you should know that before you dive into a protocol for tissue clearing. Now, the way a confocal microscope works is that we're shooting a laser into our tissue off a dichroic mirror. And then what's happening is that laser is exciting a fluorophore within our tissue that we've labeled, and that's coming back through that dichroic mirror, and it's going through a pinhole system. And that pinhole system, what it's doing is it's only allowing light that's coming from the plane that we're imaging at or want to image at to enter into our detector. So this allows us to image an infinite number of planes throughout the depth of our tissue. But one of the problems with this is that while we're only collecting data from a single, very, very thin plane in our tissue, we're illuminating the entire depth of our tissue. So the fluorophores at the top of our tissue will start to quench way before the fluorophores at the bottom of our tissue. And overall, if we're imaging a tissue for a long period of time with confocal microscopy, by the time you get to the bottom of your tissue, or the top of your tissue if you start at the bottom, you're going to have quenched all those fluorophores, and you might not get any signal back from your detector, and that creates a lot of problems with trying to get uniform imaging throughout that depth. So with confocal microscopy, that's always a consideration, is that quenching, and that's why we always suggest using a thinner tissue section for confocal microscopy because of that effect. To show a little bit more detail on this pinhole element works, because this is the entire background of what makes a confocal, is that while we're illuminating entire depth of the tissue, we're selectively only capturing that imaging plane that matches up with our objective to our detector. And that allows us to get very, very crisp images back to our microscope that we can analyze after we collect all the imaging data from our tissue. There's a lot of principles of microscopy to know, but the most important principle to know is working distance. So working distance is the distance from the bottom of your objective to its imaging plane. And that depth will be the limitation of how far you can get into a tissue. So if your working distance is 10 millimeters, for example, you're only ever going to be able to image 10 millimeters into your tissue. You're not going to be able to go any farther than that because you're limited by that working distance depth. What's really important is as you increase your magnification, generally speaking, that working distance will go down. So for a 40x objective, you might only have a 200 micron working distance that you can work with. So while you'd love to image an entire mouse brain in 40x, you then have to slice an entire mouse brain into 100 or 200 micron sections to be able to do that. So there's some practical limitations associated with just objectives that dictate how far you can get into a tissue. And then field of view. So we look at 4x or 40x, that field of view is 10 times smaller with a 40x compared to a 4x, and because of that, imaging will take 10 times longer in a plane, and will take up to 1,000 times longer in 3D because we have 3 dimensions for that 40x objective compared to that 4x objective. So field of view will dictate how long an imaging run will take for your tissue. And then lastly, numerical aperture is a number that will dictate the quality of the images that you acquire. A higher NA, the better, generally speaking, but high NA, long working distance objectives can be very expensive. These long working distance objectives with high NAs can be $10, $20, or even $30,000 to image deep into a tissue. So again, that's a reason why many researchers use inverted confocal microscopes with inexpensive air objectives and limit their tissue thickness to 1 millimeter. They don't have to buy very expensive optics just to see slightly deeper into their tissue. Now, I talked about this previously, but the reason that we're not able to see, of course, through our hand is because of differences in refractive index. So all of the cells within my hand have differences in refractive index between the proteins, lipids, and cytosol. And that's what's causing light to diffract and making my hand appear opaque. It's much similar to when I'm looking at a glass of water and I look at a pencil behind that glass of water, that pencil is dramatically distorted because as light's passing through air and then water and then through air, that light is bending and causing that pencil to appear like it's also bent, but it's a refractive index mismatch issue. So refractive index mismatch is what causes a lot of artifacts when we're trying to image deep into a tissue. So we have a tissue that's in an imaging solution that has a similar refractive index, and we have an objective that might not be perfectly matched with refractive index to the tissue and the media that tissue is in. And because of that, we will get differences in refractive index and diffraction that as we get deeper and deeper into a tissue will start to cause artifacts. So our image quality, the deeper we get into a tissue, will start to become worse and worse as we get deeper. So again, this is a reason to keep tissues thinner, so around 1 millimeter or so, which is a practical limitation for many microscopes, and also a practical limitation before you start getting very severe imaging artifacts due to changes in refractive index, specifically if you're using an air objective with deep tissue imaging. So it's very important to understand the refractive index of your microscope objective and trying to match that as closely to the imaging media as possible. Now to show what this looks like, we're looking at a tissue here and we're imaging through that tissue deeper and deeper. And what you're seeing is if we image 100 microns into that tissue, we're getting a slight change in refractive index, and that's causing some distortion. But as we get deeper and deeper, that bending of light as we go from our media to potentially a cover slip to our objective is becoming more and more severe, and because of that, the image quality is reducing the deeper and deeper we get into a tissue. So this is a really important consideration to be aware of when you start adopting tissue clearing into your research, is that while you'd love to get really deep into your tissue, it might not be possible regardless of how clear that tissue is, just because of the objectives that you're working with. So those are the details on confocal microscopy, which I would say is most common. Most researchers that we work with use confocal microscopy, and in our own contract research services, we primarily use confocal microscopy, and a lot of times we use high-content automated confocal microscopy because we can set up many, many 1 millimeter sections of mouse brain tissue or liver tissue all at once, and we can use an automated confocal inverted imager to image all of those tissues for a day or more to collect all of that data, whereas if you try light sheet microscopy, that throughput is going to be much lower, but you can image a larger volume more quickly. So light sheet microscopy, it's ideal for imaging whole mouse brains, and most of the large tissue imaging papers that you've seen use light sheet microscopy. And the way light sheet microscopy works is to pass a very, very thin laser light sheet through your transparent tissue. Laser light sheets are incredibly simple. While these microscopes are not that common, there's not a lot of them out there, and there's few commercial companies that are selling them, light sheet microscopes are actually very simple. You can construct your own light sheet microscope for around $30,000 using the open SPIM resources that are out there. And the technique itself is simple, but it's relatively challenging to get a very thin light sheet through your tissue. But the way we image the tissue using light sheet microscopy is to simply generate this very thin sheet of light and to move that sheet of light through your tissue, which allows us to capture different planes throughout the depth of our tissue, or we can move our tissue through that plane of light. But either way, we're acquiring these steps throughout our tissue and acquiring all those individual slices as we move this laser light sheet through our tissue. It's very rapid. It doesn't cause as much quenching as confocal microscopy and really allows us to get a very high level of quality through very large tissue volumes. But again, these are not very common. Sample prep is relatively challenging, and these are not able to be easily automated like you can with confocal microscopy. One of the challenges we see from many researchers is the data. So we get through labeling, we get through tissue clearing, we get through the imaging, and then we generate this substantial amount of data. And a lot of researchers struggle with what to actually do with this data, how to process it, how to view it. Many researchers are simply interested in getting some pretty pictures for publication, and that's relatively straightforward. But going ahead and actually analyzing this data can create a lot of bottlenecks for researchers, and that's one of the reasons that when we work with our clients on 3D tissue imaging projects, we stress up front that while we can acquire substantial amounts of data from a tissue, we need to ask ourselves what data is actually meaningful to answer my research question. If I use 4x imaging and that allows me to answer my research question, there's no reason to use 40x imaging that creates 1,000 times more data and will take 1,000 times as long to actually image the tissue. So figuring out up front how much data you want to generate is very, very important to allow you on the back end to actually better answer your research question. For processing the data, we typically process all the individual Z-slices from a 3D imaging campaign individually on their own using tools like Cell Profiler or ImageJ, and this allows us to extract quantitative metrics from our tissues and answer simple questions like co-localization and mapping vasculature and mapping different complex features in 3D. But that will really depend upon your research question specifically, but we always suggest to minimize that data as much as possible on the front end. Now again, interpreting this data is a bottleneck for a lot of researchers. However, there's lots of resources through Cell Profiler and ImageJ that allow you to use open source and free tools to process that data, and there are a number of publications that are out there that allow you to understand which macros and which plugins to use for specific research questions, but this varies from our experience greatly with what a researcher wants to do, from just getting a great three-dimensional rendering to try and analyze how a vasculature network and a brain works. So it really depends on your research question, but we always suggest to researchers to try and dive in first to the free open source tools that are out there. They're a little bit harder to use than some of the more expensive software, but 3D analysis software can be incredibly, incredibly expensive. So we would suggest to dive into those open source platforms first before going into something more expensive like Imaris. Now to wrap up here, we've developed several 3D tissue clearing and labeling kits with Abcam that allow you to easily adopt this into your research workflow without having to go through the process of optimizing the dilution, optimizing the incubation time, and figuring out exactly how to process your tissues. And these kits have been developed for 1mm mouse brain tissues as well as 3D cell culture models to allow you to easily adopt this into your research workflow today. Now, thank you for your time. I much appreciate it.